Alright, so we're going to be talking about basic genetics, and the next few chapters are all actually genetics-based chapters. So before the actual notes, I wanted to make a real quick comment. Keep in mind that genetics is based on probability. So it doesn't mean that if you expect 3/4 to be tall and 1/4 to be short, that if there are 4 offspring, you're going to get 3 tall and 1 short. Not necessarily. Just like flipping a coin, it's a 50-50, but if you flip a coin 10 times, you might get 8 heads and 2 tails. That's just genetic probability. The bigger the sample size, typically, the closer we get to the predictions. We used to do a lab where I had people flip coins, and we would actually flip the coins 100 times, and pretty much every single lab group consistently would get answers that, at least by a chi-square test, would pass a chi-square test with being close enough to the predicted ratios. But if they only flip the coin 10 times, lots of people would fail the chi-square test. The sizes are just too small. Yeah. So, Mendel, he is considered the father of genetics. The reason he's the father of genetics is because he's really the first one that actually used math and applied mathematical concepts to the numbers of offspring that he got. Other people had studied genetics before him, but a lot of them just gave qualitative descriptions of what they got. The common belief at the time was in what was called the blending of traits. That if you cross, and in people, I can see where people might think that's what's happening. You cross a dark person and a light person, and you get people that are medium skin tone. Or dark hair and light hair, and you get people with medium hair. So when you saw results like that, it would be easy to mistakenly draw a conclusion that everything just blended together, which is not what was happening. Mendel was the first one to mathematically show what was actually going on. Even though, keep in mind, this is the 1890s, they didn't know about mitosis, meiosis, they didn't know about DNA, they didn't know about chromosomes. Everything he discovered was just by looking at the external appearances of offspring in his cross. That's how he came up with his principles. He studied the pea plant. There are a lot of reasons why it turns out that pea plants were a really good choice for studying genetics. The first one is that they were self-pollinating, which isn't going to make a whole lot of sense other than I can explain it to you to say that if you look at the structure of a plant, they kind of look like this. You have the male and the female part. In peas, those are on the same flower. So it was really easy for him. If he just let the plant reproduce on its own, the pollen would basically fertilize the same flower. Which means that if you knew that this parent was big B, little b, and it's the same parent, was also big B, little b, you basically could do simple genetic crosses from the same flower pollinating itself. Even though it seems like that wouldn't be sexual reproduction, it's still sexual reproduction. But even better than that, because pea plants don't really move around, he could also cut this off of one pea plant and then fertilize another one. So it was really, really easy to cross whatever plants he wanted to just by controlling the spread of the pollen. The second reason why pea plants were really easy to study is that there were 7 traits that they had, and all of those traits were really easy to distinguish. Meaning, for example, if you crossed a tall plant and a short plant, all the plants came out tall. There was no medium, there was nothing in between, it was either tall or short. When you crossed purple flowers and white flowers, all the offspring came out purple. There was no pink, there was nothing in between. If any of that had happened, it would have confused him. He wouldn't have been able to come up with his conclusions. But every single trait that he studied was a simple trait that showed dominant and recessive. So he was able to come up with conclusions from that. And finally, pea plants grow super fast. They're really easy to raise. And they have a lot of offspring. Every pea in a pea pod is an offspring. So technically, you could grow a plant from a single pea, and every pea that plant made, within a month, 100 peas would be an offspring. So that made them super easy to study, because you could go through 5 generations of peas in just a couple of months and look at the outcomes. People are super hard to study. It's really hard for us to discover genetic traits in people, at least back in Mendel's time, because we're the opposite of all of these. We don't just make people to see what kind of babies they're going to have, although it would be interesting. A lot of our traits are not easy to see. You look at eye color. It's not just everybody either has blue or brown. There's all these shades of blues and shades of browns and shades of greens. And the same with hair color, skin color, height. Our traits are complicated. We don't grow fast, and we're not easy to take care of, and we don't have a lot of offspring. Every single reason why pea plants were a good choice, we're a crappy choice. And it makes it much harder for us to learn a lot of the traits in people because we had to rely on family trees going back, a couple hundred years. Oh, absolutely, yeah. Now, animal-wise, what is a good animal that you could use to study genetics? Flies is good? Rats is good? This was actually an AP question. They gave a cross a few years ago, and the first question they asked was what organism might have been a good choice for this experiment? Flies, rats, mice, rabbits, dogs, cats, any kind of insect, birds, fish, pea plants. Anything you could come up with that would make lots of offspring, be easy to grow, have a very quick lifespan was an appropriate answer. Elephants would have been a crappy answer. Okay, one baby at a time, and it takes them 8 years to have to raise that child and have another one. Monkeys, bad choice. That would not work. Bacteria, not a good choice. They're not sexually reproducing. So that's all they were looking for. Mice, rats, pea plants, fruit flies, all really good answers to a question like that. Anything that's sexually reproducing and reproduces quickly, that you could control the breeding and make lots of offspring. All right, so what he actually would do is he would take plants that he considered purebred. Now keep in mind, he didn't know they were genetically pure necessarily, but since they were self-pollinating, he could have a plant that consistently always made, for example, the example we're going to do, always made round peas. Every single time, generation after generation, it made round peas. Its offspring made round peas, etc. He would call that purebred. So he would take one that consistently made round peas, cross it with one that consistently made wrinkled peas. So here's a sample of one of his crosses, and we're just going to spend a few minutes walking through a bunch of genetics vocabulary that applies to this. So first of all, anytime he started with two parents that he called purebreds, he called the original cross, or the original generation, the P generation. That stands for parental. They're the original parents. But if you are asked to do a cross, and they tell you you're starting with a P generation, you can assume that you are starting with purebreds. A P generation always consists of purebreds of two different traits. So round versus wrinkled, or red flowers versus white flowers. But it's always purebreds in a P generation. Their offspring are called the F1. It stands for first filial. I think in Latin that means son or child or something along those lines. But this is basically your offspring of your purebreds. And then they typically will ask you to cross the F1 to get your F2. So when they say cross the F1, they basically want you to cross an offspring with another offspring from the same cross. In other words, two that basically have the same genotype. You'll see in a minute when we get to some more of the vocab. You never really go beyond F2, because when you get to F2, they don't all look alike, so they can't really tell you to cross the F2 because you wouldn't know which ones to cross with each other. In the F1, they're all going to look alike. All right, so now that's the first thing. Second thing, he discovered that every single time, no matter what two traits he picked, one trait always covered up another. In this particular cross, when he crossed round and wrinkled, every single time the offspring came out round. So he had a name for that. He called a trait that covers up another trait dominant. So he would now say that round must be the dominant trait, and what would that make wrinkled? Recessive. Good. I'm sure you've heard most of these words before. So he called the trait that covered stuff up dominant. He called the trait that got covered up recessive. You might say, well, what would have happened if he had crossed, round and wrinkled and got something in between? Well, he probably would have been really confused and he wouldn't have been able to draw any conclusions. That's the truth of the matter. But none of the traits in peas show incomplete dominance or weird kinds of outcomes. Every single trait he studied was either dominant or recessive. So this is why he was able to draw his conclusions. Now, the other thing is, he said that he started with purebred. So when you start with a purebred generation, what he did was he decided you would use letters to represent the traits. So the gene for the trait, in this case, the trait was the shape of the seed. That was the gene. It had two versions. The versions of a trait are called alleles. One of the alleles was the allele that coded for round, and the second allele allele coded for wrinkled. Now, you can choose any letters you want, but first of all, bless you, when you are looking at a single gene, you usually pick one letter. Common rookie freshman mistakes, they want to use R for round and W for wrinkled. You don't want to do that. It's one gene for flower color. There's just two versions of it. So what you do is you pick one letter, you use capital letter, so we're going to use big R for round, and then we're going to use lowercase letter for the recessive version. So we're going to use capital R for round, little r for wrinkled. So that's, now, again, nobody's going to grade you based on what letters you pick. So if you chose to use S or P or Q or I don't care what letter you use, but if you see it on a test, typically that's where they're usually getting the letters from. That's what you do. You pick the letter of the dominant trait, capital, lowercase for recessive. All right, now, he said that the original ones were purebred. He figured out from his crosses, again, something people didn't know prior to his time, that they must be carrying two copies, which we now know, right? That you're diploid and you have two copies of every chromosome. He was already proposing that for every factor, is what he called it, the parent was carrying two copies. Since this was a purebred parent, he said that both copies must be the same and they must be capital letters. So he would say that the original parent here was big R, big R. He came up with a word for this as well. We called it being homozygous. Specifically, this one would be homozygous dominant. What letters would be used for the wrinkled one? Here. Little r, which he called what? Homozygous recessive. Exactly. And it's homozygous because the two letters are the same. And he was assuming that the two letters were the same because of the fact that he was assuming he was starting with purebreds. Because they had made generation after generation, the offspring always came out the same way. Now today we could handpick and make sure they weren't purebreds. Oh, because, and we're going to learn about this actually either tomorrow or Friday, because skin color, for example, is controlled by at least 5 genes, not one. So the combinations of all 5 of those give you this range of 20 or more different shades of skin color. That's why. It's not just one gene. Any trait controlled by just one gene, the maximum phenotypes you could see would be 3 if it was incomplete dominance. You could get like black, white, and gray. But obviously people have way more shades than that. So it's at least 5 genes that are controlling it. Now, so the next thing that he figured out was that even though a parent was carrying 2 copies, they were only giving towards the offspring 1 copy of the genes that they had. This only carries big R, so he must have given a big R. This one only carries little r, so they must have given a little r. And so the offspring of this cross, all of them, would have been big R, little r. Now, they would have looked exactly the same as this original parent. That's the whole idea of what dominant is. If a trait is truly dominant, then big R, and big R, little r are both going to show the same dominant trait. You wouldn't physically be able to distinguish them. What did he call it when the letters were big and little? Good. So this was called heterozygous. Sometimes you'll also see the word hybrid for this. I prefer the word heterozygous, but I do see that word used a lot, hybrid. When I think of hybrid, I think of like, like a dog cat, like some kind of a fused hybrid creature. But hybrid can be used in genetics just to mean the same as heterozygous. All right, now we're going to talk about F2 in a minute. But the bottom line is, at the time, remember I told you that they believed in like the blending of traits, or that one trait overpowered another. Like they would have never gone past the F1 generation. So in his time, they just assumed wrinkled disappeared forever. He proved that wasn't the case. He showed in the F2, wrinkled comes back. That wrinkled technically is not gone, it's just covered up. So we're going to talk in a few minutes about how it came back in the F2. But that's a whole, in a nutshell, a whole bunch of genetic vocabulary, which is listed on another portion of this lecture. So we're going to move on, and you'll see we're going to revisit this vocabulary in a couple of minutes. He came up with 3 principles from what he studied. The first one is called the principle of dominance, which is super easy. Just said that some traits covered up others. The ones that covered up others were dominant,